Oh my god, he's finally fucking streaming. My prophet's finally streaming the fucking screen. Holy shit. For once, man. Wait. Does he have clicker on? No way. Alright, you gotta watch or at least watch the final cutscene. Because the final cutscene of the Lantern Rite is actually crazy. I think this is where they spend most of their budget. Destiny has no budget to spend, bro. Nah. Okay, but Destiny is just a fucking money trap, bro. Bro, Destiny is basically the GTA 5. And you know how long Destiny 2 has been out and they're just milking it like crazy? And it's ass. And it's still garbage. Nah, Destiny 2 is garbage. It's garbage.
Recording in progress.
Hey. Good day, everyone. So, interestingly enough, it looks like the annotation tool is working. Don't make a liar out of me. So, I can draw today. It's working. Yet new problem is I brought a special drawing board thing to make it a little bit easier for myself, but that has its own issues. It's only working on one screen and not the screen that I'm sharing with the Zoom and everything. So, anyways, so today it's a Q&A session. I'm here to answer questions you may have. We're in the evaluation phase of our deep question 1, which is due on Friday, I believe, right? Is that right? Friday? Yes, Friday. And so since we're in the evaluation phase, you can ask questions about deep question 1. I can answer those questions. I can answer questions on anything that you may, any questions you may have on the information prior to deep question 1, but the information that's on your pre-labs, like for next week, those questions I cannot answer, of course, right? So I think week 5, the pre-lab materials, gene expression has been posted. Everyone see that? Next week, you'll have your deep question 2 being released. If you have no questions, nor are you interested in hearing other people's questions, you don't have to feel obliged to stay. Enjoy the rest of your day. If you do have questions, now's your time. Yeah. Sorry, with the mask on, it makes it hard to. Yeah. Okay. Can you clarify, what do you mean the X, okay. How about, do you want me to, okay, do you want me to draw this out? I should be able to draw now. Okay, so, yeah. So you remember X, I didn't catch that part.
Where you can
Yep.
do some rolls, and you don't know what you're getting. Yeah.
Oh,
Yeah.
it's still the same thing. Okay, so if we draw a chromosome as an
But
X,
Genshin is
or we
one
draw
of
it
the
as a
more
single
favorable
bar,
gachas.
are those the
'Cause
same?
most gachas don't actually like,
Actually,
guarantee you anything.
just pause for a sec. Let me answer that accurately.
Like for Genshin, if
No,
you
they're not the
if you
same.
hit a limit, you're
And
guaranteed to
actually,
get.
the bar that you see, that bar is you see it in textbooks and sometimes on the internet as a, it doesn't
not
even
your
really
odds go up, but your
exist
like
until the
if you
X
hit
gets
like
pulled apart.
yeah.
So for a very, very brief time in the cell cycle, do you actually have a chromosome that looks like a bar? Or- Well, okay, so that's when does the X
Yeah, like
become
let's say
two
you're able to
bars?
farm for
Can
Rivens,
somebody
right?
tell me that?
Every
Is it
every 5
meiosis
minutes
1 or meiosis
you have
2?
a chance of getting a Riven, right?
That
But
the
if you don't get Rivens for
chromosome
like
that has two
a
chromatids
whole 2
and
hours,
attached
on
at
the
the
third
center
hour,
gets pulled
they
apart
guarantee
into two
a drop of a
bars.
Riven if you haven't already gotten it.
Uh,
But if you did,
Two,
it resets.
meiosis 2, yeah. Remember in meiosis 1, you have those X's and they align differently than what they would align in
That's why I
mitosis,
enjoy
right?
Genshin.
Mitosis, they're aligning like this, and they replicated, and the
Cause
replicated chromosome,
Cause
the
they don't
chromatids get
because
pulled
they don't
apart,
get enough money.
and then you
if
end
they
up
do
with
that.
identical. You end up with identical chromosomes in each of the cells
because think about it like this:
each chromosome
if you
aligned
have
head to
the
tail, head to
worst
tail, head
luck
to tail,
ever in
right?
Genshin, you have
So
to spend
that X does get pulled apart.
a
Gets
minimum,
pulled apart,
or like the
in
most you would have to
mitosis.
spend is like
It gets pulled apart
$300
in
to get what you want.
meiosis as well. At the end
That's
of meiosis,
that's just for one character.
near the end of meiosis 2. Yeah. Their end shape, there is no end because a circle never ends, right? But the shape that they're in the vast majority of the time in the cell cycle, and the shape, like after they get pulled apart, they decondense and they end up like the noodles in the bowl of soup look. And that's where they spend the most of their time. G1, S phase, G2, vast majority of the cell cycle, they're in that phase, that state. The relaxed state, they're the state that permits high levels of gene expression. Yeah. Neither of them, if there's, so neither of them are wrong unless there's something specific. Like if I was asking, well, what do you find at metaphase, whether it's metaphase 1 or metaphase 2, you find the X's. You don't find the bars. The bars only happen later, right? They only happen later, later on in following metaphase and mitosis. They get pulled apart. Following metaphase 2 and meiosis 2, they get pulled apart. And so my answer to that would be neither of them are wrong. It's just both of those only exist for a short
This is
time.
the first time they actually did this shit.
And really, the bars, the individual bars, rather than the X's, the individual bars exist
Because it's offbeat.
Even less time, right?
It's because the original audio is Chinese.
Oh no! Well,
Yeah.
no,
Like
no.
the beat is made for the Chinese lyrics.
Generally not. If you by time you mean like for a brief second, or like maybe one broke apart and the other one's just starting to break and the other one's already broken apart, like maybe for a brief instance,
Yeah.
but no, generally not for long, right?
Oh, little kids, bro. Sure.
You know, okay. One of the things I really would like to explain to everyone, and this is it kind of trips you up, but it's a really important point, and it's a point that people generally are not willing to accept.
Jeez.
An important point that everyone should know is that all the things that we describe in biology and classes and stuff like this, none of it is absolute. Almost everything has exceptions. So she was asking me, does the bar on one homologous chromosome ever exist with the X on the other homologous chromosome? Generally not. For a brief instance, possibly, if one of the homologous chromosomes got pulled apart more rapidly than its homologous partner did, yeah, maybe. But generally, you would not see an X with a bar. Okay. Yeah. There's a lot of.
Yeah, he's a god.
You know, this section, I think after it clicks, it's kind of like, for me, I was always bad at math, and not bad, but not great at math. And I had a hard time with Calc 1. Who had a hard time with Calc 1? Just me? Okay, there's one or two. There's about 4 or 5 people admitting it. It was really hard, and I really struggled to pass. Actually, I took Calc 1 twice. I'll admit. Here's your prof admitting he took Calc 1 twice. First time, just couldn't get it. One day in class, it just clicked. Wow. And I just understood. I don't know what happened. I just understood. And second time through, I got an A+. I had to really work for it, but I almost failed the first time. I think with the chromosomes and their segregation, and I'm not bragging here. I'm just trying to make a point. Chromosomes and the way that they segregate and how alleles work and everything, it can be really, really confusing. It's kind of buried information. And so you try to study it and really try to draw things out.
What an amazing story.
People don't want to do it, but draw it out. Look at it with a friend.
It only
if
happens
you want
once
me
a
to
year,
look at
bro.
your work, come and ask me. I'll take a look at it. But it's one of those things that you kind of maybe don't fully grasp.
No,
and that's the reason why I say that is because I'm going to ask questions. There's going to be questions about this kind of stuff. You're going to see it on Deep Questions or whatever. And I still see a lot of people are not getting it. It's just one of those things like it actually takes work and it takes some real thought to okay, I finally got it.
Yeah.
If you want to look at some chromosomes and stuff, we were talking about differences between mitosis and meiosis. My pen's working.
Wait,
I'm all excited now. I can draw some stuff over it. Whatever you guys want. I'm here. I'm at your disposal. Whatever you want me to do. Yeah. Last session, I got the slides up, so let's check it out. Last iClicker question. Good. Okay, it's probably this one you're referring to. Let me know if it's not.
Wait, what is this?
Does one have less than one? Let me see
Oh.
here. No. No, this one. This one. Yeah, okay. Yeah, this one's important. Very important.
Yeah.
Yeah, because sometimes I get off on
That is actually the
tangents, and then
best.
towards the end of the class,
4-star.
I start speeding up. I'll try to regulate the pace. It's because I want to give you guys a chance to draw things
Oh,
out
you
and
have to
stuff,
roll
but
Bennett.
then we have to try to cover everything. But this is great. So we got a Q&A session to go over this. So let's do that. So for genes A and B, so we're talking about these two genes here.
Yeah, that's one of the
Nero's
femboys.
genotype is A1,
Yeah.
B1. So that would be A1, and this is going to be B1.
I'll show you the real femboy.
Okay. Over here, what are we going to have over here? A1, B1 again. They're identical, right? Because the DNA is just
didn't say that.
replicated. The X comes from the same DNA. Sorry, A1. That's why I was hoping my pen could work. It's a lot easier. It says B1.
You know.
Yeah. And then over here,
Some offbeat rap.
this is the other chromosome. We got A2, B2. Okay. A2, B.
Big Dong Zhang.
Not the best drawings. Okay, so Nero can produce about 1 million gametes during their life. Okay, I thought Nero was a person, but anyways, during life. If genes A and B are immediately
King
next
Shui.
to each other, so that's important information. They're next to each other on the
Shang.
chromosome. How many of the 1 million gametes would you expect to be recombinants? All right. Okay, so let's look at the possibilities, right? But we're talking about recombination here. Let's assume we're not talking about, okay, so it's, yeah, okay. Ultimately the question is, how often are you going to find recombination? Because it's wondering, another way to ask that is, how often are we going to find A1 ending up on the same chromosome as B2? Or A2 ending up on the same chromosome as B1? So we need to have a crossover happen. We need to have a recombination happen. All right. So now I think the confusing thing is like, how do we get numbers out of this? And how do we know that it's going to be less than half? Because we said that it was less than half. That was the answer, right? Less than half of the time. Okay. I want you to take a step back for a second here and think about A1 and B2, okay? If they were, they're on different chromosomes. How often are they going to, let's say that if they were on different chromosomes, never mind the same chromosome for a second, they're on different chromosomes. Okay, so I'm gonna draw new chromosomes here, there. And I'm gonna put my A1 down here. And I'm gonna draw a notably different chromosome here. And put my B1 on it. And of course, this is gonna be a diploid organism. So let's just say like this, A2, and over here, B2. I have to figure out how to get my pen thing working on this screen. I'm gonna have to, I know I looked it up last year and there was like 100 people, blogs and everything, and nobody knew how to do it. Okay, so if they're on different chromosomes, what proportion of the time would you expect A1 over here to end up in the same cell as B2?
Oh, you told me to shut up.
Okay, So let's think about this. Let's think about this. In mitosis, how do these line up? They all line up head to tail, head to tail, head to tail, right? This chromosome with A1 here is going to align head to tail with its homologous chromosome. So actually, it's going to look like one on top of each other.
To be honest, it's actually
Here's
pretty
our
good
A1
to other dialogue games.
And then there is our
first spoken.
our A2, and then our B's, and so on.
But the
so
Japanese,
now
they actually spend bank on that.
The DNA gets pulled
Yeah, you know,
into
you know,
the DNA from these chromosomes.
The
it gets separated, right? This is our A2.
You know, Kirito?
It gets separated, split in half. Half is going to go
He took
to
his voice
one
actor.
cell.
They hired him for him.
Half is going to go for the other
And
cell.
do you know?
And that's the same thing happens for A2.
Did you
The
ever
same
watch,
thing happens for B's. So if you have your B's down here,
Konosuba?
this is your
Konosuba?
B1. This is your B2.
Or have you heard of it?
How often would you expect an A1 to end up in a cell with a
have you watched
with a
ReZero?
B2? This is mitosis.
You know Emilia, the main girl? That's Su Tao. Yeah. Megumi, the exorcist.
Hold on.
bitch.
I'm kind of, I'm putting this is not to be tricky, but I'm just putting this out there.
And you,
I want to cover everything, right? So in mitosis,
you know,
the cells end up being genetically identical.
That's Zhongli.
Right? Genetically identical.
Every,
okay, so when they line up like this, you got your homolog one on top. The DNA was replicated, so it's making its X form with its chromatids.
Demon Slayer.
And remember, half of this chromosome is going to go. It's going to go to one cell, and half is going to go to the other cell.
Like that, just spent bank.
Half of this chromosome is going to go to one cell. Half is going to go to the other cell. And then ultimately, you end up with what you started with before DNA replication occurred. You just end up with what you started with. Okay, that's in mitosis. Okay, this is in mitosis that happens. Mitosis. Ah, oh my gosh.
Palmer just sounds gay, but all of them.
Now in meiosis,
that's
the
a
chromosomes line up on the plate like this. In metaphase.
If you saw that, you would think that's a female, right?
And remember we said whenever they line up like this.
That's a male right there.
This is meiosis.
Peak male physique.
I'm just gonna like this. Okay. They line up like this. We said that the homologous chromosomes, you don't know which side the homologs are going to align on. They align differently. It's not head to tail, and they're going to align on each side of this plate, right? And so the A1 can go on this side, and either the B1 or the B2 can go on the same side. All right. So that means that A1, if they're on different chromosomes, A1 would end up with B2 50% of the time. 50% of the time, A1 would end up with B1. Okay, now, whenever the genes are linked, it's a different story. Okay, because you have A1 and the A's and the B's are on the same chromosome. Okay, and so here we say that Nuru actually has A1 with B1. Okay, so to get a recombination has to happen to get A1 with B2 on the same chromosome. It has to happen right here, right? You'd have to have your recombination like this, which is shown here. That's what these are. You can see here that this is your B1. Anyways, so this is actually B2. And this is over here is your B1. So the question is that, if genes A and B are immediately next to each other on the chromosome, okay, how many million gametes would you expect? It doesn't matter, never mind the million part. How many proportion of gametes would you expect, okay, to be recombinant? The fact of the matter is,
Look
Is
how
that
short those shorts
since
are.
the genes are close together, as indicated, they're immediately next to each other. Oftentimes,
and then you have
this most
guy.
often, this A1 allele is going to segregate with this B1 allele,
Huh?
right? And
Yeah,
you're
it's a
not
five
going to
cell.
have this recombination that they've represented here. And so
This
you're
guy?
just by chance, the segregation, like if they
There's a
were
new one.
unlinked, it's 50%.
This
This is
guy.
how people detect it. They actually do
This guy
crosses,
is
and this
in.
is how they detect linkage.
No,
in real
that's
life.
not the fembar. I'm saying
Research
the very
is they
first
do
male
crosses, and
was a
then they
femboy.
have their parental combinations, and then what they
That's
do
the
is they
second male.
look
Yeah,
for,
this is the first ever male.
rather
which is
than
a
getting
femboy.
a 50-50, I'm getting this phenotype segregating half the time with this version of this
I'm pretty sure
gene, this phenotype. I'm seeing that the parental phenotypes are segregating with each other more so than I'm getting a new mixture of phenotypes.
Hmm.
And that tells
I
you
wouldn't be
that those
surprised.
particular genes are linked. It's used in mapping, which I don't know why in this course we don't cover it, but I think you guys don't need to worry about it, but I think in future years, I'm going to talk to the other profs that teach this course. I think mapping is important. It's important to know how to map genes,
Like what?
but anyways, that's how mapping is done. And that's how distances between
they're not
genes
the
is
main
figured
source
out.
of
Because
stuff.
the genes that are linked on the same chromosome,
get
the
stuff
closer
through
they
a
are
allele, or
together,
even
the
just
more
straight up by
the phenotypes are found to segregate. The parental phenotypes are found to segregate with one another. The further they get apart, but that's, so the further they get apart, the more you'll see recombination, and the recombination introducing the new combination of phenotypes, like the phenotype that comes from A1 and B2.
Almond tofu.
Now, so it can be mapped, the distances can be mapped using math. I would like to get into that, but we don't do that this year. But anyways, and so that the distances can be
How
mapped
long is this
until the
fucking
genes
quest
get
line?
far
Oh
enough
my
apart
God.
that they no longer are found to be linked. So even though they're on the same chromosome, they may not segregate together because
Like the
when they
battle
get
pass
further apart, that's kind of like the point I was talking
it is.
about the small dartboard versus the huge dartboard, right? The further they are apart, you're going to have the synaptonemal complex forms between these chromosomes, and you have crossing over happening all along the length. It doesn't just happen once. It happens, I'm just drawing a few times here. It actually happens, can happen thousands of times. It depends. People may say, like, how often does it happen? There's recombination hotspots. There's recombination spots where recombination doesn't happen very frequently. It's all based on the protein composition. It's all based on the ability of the synaptonemal complex to set up. But anyway, so if you have a big, let's say the A was all the way up here and B is all the way down at the tip.
Ew.
They may not even appear to be on the same chromosome. In other words, that they would still show this Mendelian segregation, not a linked segregation where the alleles are found in higher proportions, like the parental alleles are found in higher proportions.
Look at the stuff I can get. I can get a 4-star weapon.
If they're far enough apart, you'll get recombination back and forth, that essentially they're not really linked. Those alleles switch up all the time, and you never would know that they're on the same chromosome. Long answer, I know, but I think I covered a lot there that I wanted you guys to know.
680 divided by 160. What is that? Yeah. That's like 5?
Yeah.
4?
I see.
You have 4 rolls. Generous amount, bro.
Oh, okay. So explain that again. So you're right. So only 50%, even if there was recombination, only 50% of the gametes would get the recombinants, right?
Generous.
But then the answer would have been A. It would have been 50%.
The Mora, like the currency, is you actually get a lot.
Ah. Okay. Fair enough.
Yeah. Yeah, I
Fair
have
enough.
58 mil.
But it's based on an assumption. Who knows?
Dope.
Maybe some
That's why I stopped
loci
buying it.
have such
Yeah, I bought it
recombination hotspots
every
that
month.
recombination always occurs. I would just put that out. It's possible.
Yeah.
Nature's
only-
fascinating.
2 years.
But no, that's a good. You have good reasoning.
20.
But
24.
the reason why is that genes that are close together, they generally there are alleles that are close together,
'cause-
two different genes on a
bought
chromosome.
the
They
$13
generally follow each other in segregation, and that's why you get
Because other than that,
higher
you get the money that's kind of worth it. You also get,
level of the parentals and a
this
very
thing,
low
which is
you get
the
a high
purple
level of
ball,
parentals
which
and a
is
low
another
level
pole.
of recombinance
So you get
because
like 10 poles.
recombination rarely happens between close genes. Does that make sense?
Yeah, it's a scam, but we move
Yeah.
on. We move on.
My drawings are wacky. I'm gonna work on getting this.
Oh, I'm not stingy when
pen
it comes-
installed here, but yeah. Okay, so
Yeah, bro, like, look at this. Wait, hold up. Like, look at this.
anything
Oh
else?
my god.
No, you guys are all gonna get A plus
Yeah,
on
look
the
at the
next deep
regular
question.
one,
That's
And
fantastic.
look at the new.
You guys
new
all, I
outfit.
think you did very well on the first deep
This
question.
one? Hell no. Hell no.
Well, if you
Look at this.
didn't, you got to remember
And you
that
also have-
in this course there is no final exam,
And then
right?
where's Lisa?
But there is
Like, look at
take
this.
home deep questions,
versus this.
and they do go back and cover materials that, particularly if you think they were
Oh no, this one
the
actually
class
better.
didn't do well,
The original
those will
was kinda
generally be revisited because we want you to understand those things.
But yeah.
So.
Plus
Oh,
my polls.
yeah, just one thing I'm gonna put out there. The WhatsApp group, or maybe there's probably more than one of them, but the WhatsApp for
Probably
the
like-
class, we found a lot of
600?
consistently wrong answers for deep question one.
Yeah.
Now, I'm hoping and I'm assuming that it's not like plagiarism and stuff, but I haven't heard anything about
Crazy.
that. I've just been notified by the TA.
Well, I haven't
that,
spent anything.
a lot of people are getting this,
like a whole year.
some of these questions consistently long, and they have the wrong logic.
Hey man, it was the only
It's
game I was
fine.
actively
It's great
playing.
to have a WhatsApp or whatever where the class can
No,
chat and
no.
stuff about things.
Destiny's actually
As with last year, you have to watch out for
bring me
the.
back.
I guess I don't know who's leading the discussions or whatever, but oftentimes we found very large portions of the class getting the deep questions wrong because they have the wrong logic. So I guess take with a grain of salt what you get from the your forums, your WhatsApps and whatever else. It can help you with your grade. It can also bring you down a rabbit hole and reduce your grade. So, yeah. Okay. I think you guys are expecting this to be professor-led. You're not used to having it being a student-led thing. I noticed that with my plants labs too. It might be a side effect of the pandemic and working remotely and stuff like that. You guys really want to try to be active. And I don't mind either way because I can work up here I got all kinds of stuff I can do. You want to come with questions to these things.
But there was
I'm sure there's some things that need clarification. Yeah.
Which one was that?
Yeah.
Oh yeah, I don't think so because I don't think I was high enough. Like,
Yeah.
good shit.
That was particularly the question that I think the WhatsApp group got lost on, unfortunately. Yeah, can you have 0%? Let me just bring up the question so people know exactly what we're talking about here, because I think a lot of people will forget. Here we go. Kind of thought that we would have some questions about this. Okay. Okay, question 3. And so we taught, this question asks you both about penetrance, and it asks you about variable expressivity. And I think your question was, can you have 0% expressivity and it still be considered variable? Is that what you were saying? Yeah. Okay, variable expressivity. Let's discuss what that is. So if you have no variation in the pigmentation, for example, would this be considered variable expressivity? No? Okay. Yeah, I think you're right. Yeah, so it wouldn't be considered variable expressivity. Variable expressivity is like you have a particular genotype, okay? And it's the same genotype, right? But in different individuals, you see a different range of how much that phenotype is expressed. Even though they have the same genes, the same genotype, you see a different range. So here we're saying like faint to strong pigmentation. Some of the soybeans have faint pigmentation. Some of them have strong ones. Guess what? I mean, I think it's the same thing like in CVD, cardiovascular disease. Even though the example was about soybeans, it's the same kind of thing, right? That people with the same genotypes show differences in how they express cardiovascular disease and the extent of which they'll expressing cardiovascular disease. And a lot of people said the answer to that question, well, it's probably like there was like a difference in the environment, like some of the plants had warm environment and some of them had cold environment, and that's why it had nothing, we didn't say anything about there being a different environment, right? Yeah, so variable expressivity, you get different phenotypes with the same genotype, and there could be many, many reasons for that, even under identical environments. Like I talked about the clones, right? I take a clones and I grow them under identical environment. Why they look different? Well, yeah, that's what we're going to talk about a lot in this course, is all of the things that ultimately can lead to a different phenotype, even though you have the same genotype. And then I think a lot of people, yeah, but actually I think overall the class did very well. There's still, like, I think one of the TAs was ill, so we're a little bit behind with marking question 3. So she's kind of catching up now. The first 2 questions were, they answered almost, she's almost done grading them. And yeah, the results are pretty good. Yeah. Oh, you mean are the two, okay. Can you have complete penetrance and variable expressivity? Yes. Okay, like you were saying yourself, right? Penetrance is a yes or no thing, right? It's not like expressivity, which is like a range. So penetrance is like you're looking for a phenotype, and if you don't find that phenotype in all of the organisms that have the genotype, then you have incomplete penetrance. So not all of the individuals are showing. It's just yes or no, right? Yeah. So that's like at the population level. So you could have, for example, yeah, for sure. So you could have 100%. This is what happens when the eye locus is not present. So the eye locus, we got a lot kinds of confusion about microRNA. Whoa, but we're going to come to that later. It encodes a microRNA. And microRNA silences a gene that's involved in making the pigment. Okay. And whenever the eye locus is in its recessive form, it's not causing silencing, and it's allowing that pigmentation gene to be expressed. So in other words, a microRNA is not working. It's not silencing, right? That's when the eye locus is in its suppressive form. All of the offspring would show the pigmentation phenotype. So you would have complete penetrance. But the color can be variable. Like, for example, if there was cold weather, you'll find that some of the soybeans are black color, and some of them are more of like a purple or blue color. Okay, so in other words, you have a variable expressivity of the pigmentation. That's a different degrees of pigmentation. With black being the most extreme, and then down in they can be like red. It depends on the underlying genes, and we're going to talk about that later. But if it was red was the underlying color, accumulating to the maximum, which is what happens in most, would result in a black phenotype. But with the sometimes in certain environments, the genes are not expressed well or whatever, and you can actually have a reduction. Sometimes it could just be genetics and epigenetics, which we're going to get into later. Yeah, good question. So you're really thinking about this. That's good. That's good because you want to develop your understanding. This whole course builds like you may seem like some of the sections are disconnected, but ultimately we're expecting in the end to put everything together. Yeah. All right. So does anybody want to take a shot at how would you define a gene in your own words? There could be a lot of different answers here. And there was we did have that one, was it a Nature paper that we linked you guys to that you could see how even the scientific community was struggling with this. So the point is it's very difficult to define what a gene is. More and more and more, more and more we get kind of confused. Not confused. We get exceptions to all the rules that we are building our foundation on, that we call molecular biology. Genes code proteins. Yeah. Genes only code proteins. I think Mendel got it. Genes don't always only code proteins. This is not true. Genes don't only code proteins. Actually, I think Mendel got it right. And then we thought, we know better than Mendel. We know what a gene is. You know, Mendel's saying, okay, you know, like these things that we ultimately call them genes, but it's like a heritable trait, something that codes for an heritable trait, and it has it's inherited in certain proportions and everything. Like that was closer to the truth than just saying that genes. We know that genes code proteins. Genes are a lot more than that, right? Okay, let me put it like this. If genes are just coding proteins, what about a ribosomal RNA? Is ribosomal RNA not a gene? Oh, well, there's an exception. No, there's more exceptions than there are proteins. Does everyone follow what I'm talking about right now? Some of them are paying attention. That's good. So what is a ribosomal RNA? Is it a gene? Can anyone answer that? Yeah. Is a ribosomal RNA, you know, a rRNA, is that a gene? Oh, why not? Ribosomal RNAs are not inherited. Okay. So they're encoded by the genome, right? Everything is encoded by the genome, otherwise it wouldn't be there. So ribosomal RNAs, even though we say ribosomal RNAs, they're RNAs because they're transcribed from the genome. And so there's genes there. I guess that would say that anything that's not a protein is not a gene. Ribosomal RNAs are not a gene, so they don't encode any trait. We do, but that's not the point. Like ribosomal RNAs are we inherit the genes that make the ribosomal RNAs. I would say, yes, we do inherit ribosomal RNAs themselves, but that's we're getting beyond the basics of molecular biology here. We do inherit when a sperm with its ribosomal RNAs fuses with an egg, it lets its ribosomal RNAs go into so we inherited it. Are we going to carry it for the rest of our life cycle? No, but with the DNA we will. And the DNA always encodes the ribosomal RNA. So is the ribosomal RNA, is it not encoded by genes? Is it not a gene? Not the RNA itself, but I mean the DNA that encodes it. Is it not? There's a gene that codes that, no? Yes. Okay, remember how we even got here in history, right? Is like Mendel's not the only example, but Mendel's like the famous, the P experiments and everything really did work out a lot of the math went behind inheritance. And these inherited factors that early work showed, we didn't know what they were. We didn't know if they were proteins, RNAs. I don't, like, we can make up other things, metabolites and so on. We didn't know what they were. We just knew that they seem to be inherited in certain ways. When we get this, like this color of seed, okay, it's inherited in a certain way. So something that's being inherited makes that color. And later on, experiments showed, well, guess what? Like you can actually take DNA out of one microorganism and put it into another, and that microorganism now becomes virulent, like it's the organism that you took the DNA from. So DNA ultimately encoded this gene, this heritable factor, right? Okay. And so still didn't need to know what it was, didn't need to even know it was a protein that it encoded. It's just like it's an inheritable factor, and it's a factor that contributes a certain phenotype. In that case, virulence. So then if you think about it, well, if you have a microRNA that can actually change the phenotype of an organism, it could take it from having a black seed coat to all of a sudden it's a yellow seed. Is that not a gene that does that? Huh? It changed the phenotype. Yeah. It's a heritable factor, so anything that inherits is going to get the phenotype that comes with the microRNA. So, microRNAs, it depended on how you defined a gene. One of the things we absolutely wouldn't accept was that genes, it's not a gene because it doesn't encode a protein. Well, that's not true, because not all genes code proteins. You'll be surprised to find even later on that not all genes encode RNAs. Some genes act at the DNA sequence level by itself. They don't even need to be transcribed. We're going to learn about that. So the idea is that, genes are heritable factors. They don't need to encode proteins, and a ribosomal RNA is a great example, because guess what happens if you knock out a ribosomal RNA genes? Death. You couldn't survive without proteins being synthesized. There's so many proteins that we need. Ribosomal RNAs are involved in translation, right? They make up part of the ribosome. Yeah. Okay, so you had to define a gene, and just if you, that's the one thing you couldn't say, is that, genes are DNA that encodes RNA and it makes a protein. No. That was old. That's what your traditional textbooks would have you learn a decade ago. It's not what we understand today. Okay. All right. Did anyone have a question about this one? Yeah. You were correct, up till you say, and regulates biological processes. It doesn't have to, it can. But yes, so, right. So, I couldn't even repeat that if I tried, but can you say it again? Heritable factors, okay. That control. Is that from the article? Uh-oh. Be careful you don't plagiarize the Nature paper. I don't know if the team, but yes, I think you're very much, that's very good, very good. I like it. Yeah, heritable factors, right? Heritable factors. They don't need to be proteins, and they're actually going to they're going to control, like they give rise to phenotypes. They can do that by controlling molecular processes. They can do that independently by being the trait. So carotene, for example, like the proteins you can have a gene that codes a protein, and that protein itself directly causes the phenotype with no further control or manifestation. It just directly causes a phenotype, right? And there's another layer. You can have a gene that codes a protein, which is an enzyme, and the enzyme changes the metabolites form, and the metabolites make a phenotype. Yeah, that's also an example of a gene. You can have a gene that encodes an RNA, microRNA, that regulates the expression of an enzyme coding gene, turns it off. And now it just caused a new phenotype that's also a gene. So I'm not saying that you may have what happened there? Oh, you may have said something different. It's not what I say is absolute. As long as you explained yourself well, you could get decent art marks, okay? You couldn't get full marks for saying genes encode proteins and that's the way it is. No, that's not. We know that it's not that way, and that's what all the material was about in this section whenever we describe what a gene is. Okay. Yeah. So let me see if I can pull that question back up again. Oh, maybe not. Okay. I guess Cradmark kicked me out. Okay, so I'm not going to really, I didn't want you to, one of the things I think I want you to learn is how to eliminate extraneous information, right? If you didn't know, you didn't need to know what a microRNA is even to answer that question. You didn't. Remember Mendel, right? Mendel didn't care whether it was a microRNA or a protein or what it was, just Mendel knew it's a heritable trait, it was a heritable factor, that causes this particular version of a phenotype. Okay, so not even knowing the molecular details wasn't even important. Now, you are going to learn more about microRNAs. It's one of my major complaints to the other profs teaching this course and previous courses was that we don't really get into the RNA silencing world enough, and students seem to miss it. And then I get students coming in to do research with us, and I think, like, how is that possible? Because that's it's operating all the time. I mean, it's very important for humans' evolution. It's microRNAs. There's some key changes that happen in evolution between us and primates. The mutations that change the jaw muscle structure that we used to have very strong jaws for grazing and stuff, mutated and relaxed the jaw muscles, move where they're positioning and stuff, allowing our frontal lobe of our brain to grow bigger, becoming humans, intelligent humans. That's microRNAs that changed to allow that to happen. It's not just something that makes a color in soybean seeds. Guys, this is genetics. Everything we talk about it's operating across kingdoms, across, yeah. Yes. Yeah, that's a great question, right? Does every gene have a phenotype? And I would even make it harder on myself. I'm going to go beyond that. If every gene doesn't have a phenotype, does that mean it's not a gene? Not every gene has, we mutate genes all the time. And yeah, you're right. Sometimes we'll mutate a gene and like, not really seeing any difference here. And it's kind of a letdown when you're in a research, kind of context, right? Because you want to find changes, and sometimes it happens. There's no change. Now, be careful. There can be many reasons why losing a gene doesn't cause a phenotype. It could be that other genes actually, they fill in for that gene when that gene is lost. So in other words, there's overlapping redundant functions, functional redundancies, we say. So just because you lost the gene, let's say the RNA encoded an RNA, and then that encoded a protein, and that protein is doing something. It's transporting a metabolite or something like that. That was the gene I was studying that this happened. It's a metabolite transporter, and mutated it, nothing. And it's interesting because you would put it under certain environmental stresses, and all of a sudden you see, aha, there is a big difference in the phenotype, but not in all environments. Like not when the plant is happily growing, or whenever the plant was put under a stressful environment, immediately it's semi-dwarf. It's half the size of the wild type. It can't grow properly. Stressed out, all producing all kinds of pigments to protect itself from reactive oxygen species and so on. It doesn't mean that it's not a gene, even if you looked at all the environments, like it's, when we say it's a heritable trait and it causes a heritable trait that contributes to a phenotype, you can lose it. Functional redundancies can mask it. You could lose it and maybe other genes that aren't functional redundant that can compensate for it. But the genes are usually, they're doing something. Okay, I think that whole functional part of the whole definition of what is a gene is essential. So I hope I'm answering your question there. The gene has to do something. It can't just be a stretch of DNA that doesn't do anything. Okay, so it has to have some functional, it's just, you may not see it a lot of the time. Yeah. No, and I think I was getting to that and then I just, I got off track. Yeah, so a phenotype doesn't need to be, we think of phenotypes as things we can see, we can readily detect with our naked eye, right? But a phenotype could be, it could be the measurement of a metabolite that we don't see. That doesn't cause a change in visible phenotype. I mean, if you think about like a lot of diseases are like that, right? I mean, like we don't see the disease per se, we don't see what's wrong, that a certain protein is not being put into our cells. We see later the outcome, right? But we don't see, but that's a phenotype. If you're measuring the protein levels and the doctors, whenever they do your blood tests, they're looking for proteins. Those are phenotypes. If you've lost a protein, that's a phenotype. The example with the ribosomal RNA was just to express that genes don't need to encode proteins, right? Sure. Yeah, well, and you would see it on a gel. You run it on a gel. Yeah. You could even detect it with fluorescent probes, for example, that kind of thing. Sure. And if the, yeah, so, but the point is getting back to, sorry, I don't know, what is your name? Hazad, what Hazad was saying is like, what we're talking about is like, it's functional. It's expressing a product, right? And that product, it has a function, okay? And so the mRNA, you're measuring it on a gel and then, this individual doesn't have or has less or something like variable expressivity or something like this. That's a phenotype. That's a phenotype you can detect only on the gel. An mRNA, that ribosomal RNA rather, it's gonna have a function. It's gonna do something, does something. It wouldn't be evolutionarily conserved if it didn't have a function. And that may bring up the question, well, what if it, like, this is a new protein? You have a gene duplication, two parts of a gene come together, they make a new gene. The product, as far as we know, it doesn't do anything yet. Is that a gene? Well, it's expressing an RNA, and the RNA is making a protein. The protein is a phenotype. It's presumably it's going to have function eventually. Otherwise, it'll be eliminated over time from in evolution. It won't be present anymore. No, the RNA can be the phenotype. The RNA can be the phenotype. Yeah, I know it's anything that you can measure, that you can detect, can be the phenotype. In the traditional sense of what we normally think of phenotype, it's just like what we normally think about it as a gene. It doesn't fit that traditional, but in biology, molecular biology, and science, that's generally accepted to be the case. Okay, but so you're saying, what is the genotype? So genotype and phenotype are two different things, right? Oh, no. No, the genotype is specifically, what alleles are present of a specific gene on the DNA. Genotype is specifically a sequence thing. Okay, and genotype specifically is like when we say the genotype and we talk about a particular gene, we're referring to, those pairs of, it could be different DNA sequence, but the same gene, slightly different sequence, but same gene, right? That's the genotype, gene specifically, the sequence. And then phenotype, anything I was saying earlier, so you guys can get you can already start to get thinking about this, but sometimes DNA is not even expressed. It still encodes a gene. How does it do that? They're finding relatively recently all kinds of chromosome 3D maps. And so like a DNA from one chromosome can associate physically with DNA of another chromosome. And just by virtue of its sequence, it's not transcribed, it's not translated, it's just a piece of DNA, but it can act to enhance the expression of a nearby gene. It's outside the box of what we normally talk about and what mostly we're going to talk about in this course, but you can imagine it's required for the full expression of a gene. If that DNA was mutated, now you lose full expression of the gene that it was interacting with, and you could have a disease phenotype. it's a gene. It's a gene. It's a piece of DNA. It's heritable and it has a function, right? And a regulatory function, as we said earlier. Well, I think we're almost out of time, aren't we? Oh, that's still another 10 minutes. It's good to start thinking about this. So, you know, just, you got to kind of open up your way of thinking. I think that this is one of the things that professors, we really struggle with as the students, like, you know, you come out of high school and start going to university and you've always been learning things are a certain way. And we're discovering all the time, especially in this field, in genetics and molecular biology, that things happen. A lot of what we learned before, they do happen. It does happen like that, but there are so many exceptions to the rule that having a narrow view of things is just not, it doesn't make any sense anymore. And that's what this whole section was about, right? What is a gene? It's not just something that codes a protein, a piece of DNA that codes a protein. It can be many, many other things. Yeah, there's a lot of weird stuff out there. You'll see the more you get into more and more senior courses, like, yo, that's interesting. Okay, so we didn't really talk about these much detail. Once you get this back, we can always revisit it in a future Q&A session. If you want to discuss some points, if there's something you didn't understand, you got to remember that the TA is going to do the best they can to let you know if you lost marks, where you lost marks, so you can improve your understanding and so on. They can't write out a lot for every single student because there's so many students. And so, but they'll do what they can, and usually they do a pretty good job. You can always ask if you have additional questions. You can ask me in Q&A sessions, or you can ask them. But we'll also give general feedback. So as soon as this deep question's in, I've already started putting together feedback that they've given me, but we'll give general feedback as well, more explained than what the TA can give. And if still at that point you're, well, I should also say, if you think, you know, I got this with the first critic assignment, some people, I don't like how it was marked. I don't think it's fair. Okay, I'm just going to tell you. You have the opportunity for your deep questions to dispute your grade. However, I'm not going to advise. I'm not going to discourage it. I'm not going to advise it. And just think about it like this. I'm a TA. You're a TA, right? You have 300 things to mark. And you found like a general level that you're comfortable in your grading, that you're going with the expectations for the class. So you're going to grade everything accordingly. Sometimes, sometimes there's going to be an error and it legitimately needs to be addressed. You need to draw the TA's attention like, hey, you said I was over the word limit and I wasn't. That's a clear, easy yes or no, right? But if you are saying, well, I think I did better, and you can't just say that. You actually have to give specific reasons why you're asking for a regrade. And then the TA is going to look in detail at your submission. More detail likely than they have the first time, because the first time they were doing 300, the regrade, they're going to be doing like 10. And they're going to look at it in more detail. Chances are, I don't know, I'm just guessing, they're going to be looking at it more stringently. And if anything, your grade can go down. It can go up. It can stay the same. You take the chance when you ask for a regrade, but there's no like after the regrade happens saying, oh, no, I didn't want like it gets regraded. It gets regraded. Okay, so that's the way the course is. That's it's fair. I'm not saying the TA is not going to be fair. They are. And actually I spot check and I just make sure I spot check, and they ask me for advice on sometimes whenever they're confused. So chances are I've seen a lot of what's out there. Yeah. Yeah. Yeah, absolutely. I can in the time that I have. Yes, I got 5 minutes. I can do that. Yeah. So I'm going to try to draw it out. Ooh, everybody's like, no, don't draw. Hold on a sec. Yes, I will explain. So the question is, why do we say when genes are close together that recombination happens less frequently, right? Yeah, and that's, and really you want to understand that, because that kind of understanding that concept makes everything else we talk about recombination, inheritance, and much more understandable. So hold on a sec here. Okay. So we have our chromosome like this. Here's our centromere. Yeah. Just for simplicity, I'm gonna just draw, I'm just gonna draw this homologue with the other homologue, okay? Normally,